I would like to explain how P53 is regulated. It is a protein that is very important for keeping the cellular integrity, and it is a protein that needs to be triggered. But it needs to be triggered very quickly after a damage has occurred, so it's important that you don't need to transcribe the gene and to translate the protein in order to actually have an accumulation of P53. The cell needs to react more quickly than this, so it is important that the protein is readily available to produce its effects as soon as a damage occurs. We have seen in the previous video that any damage from very cytotoxic, like lack of oxygen, to very genotoxic, like a damage to the DNA, will all trigger P53 buildup. So it is important that the protein is already available so that you can actually trigger the response quickly. So P53 is produced all the time. In fact, the authors of the book, they call it the feudal cycle. So it means that in fact P53 is being produced all the time in the cell. Some of the other proteins will be able to persist in the cell for many hours and sometimes several days. It is absolutely not the case for P53, which is in contrast degraded in about 20 minutes after its production. The cell is constantly wasting energy to produce and build some P53 subunits. Remember that you need 4 subunits to produce a functional P53 complex. But these subunits were constantly made and very quickly degraded. First of all, the complex makes a transcription factor. And this transcription factor will recognize consensus sequences in the promoter of target genes. We have seen before that we have probably more than 1,000 sequences that can be recognized and eventually activated by P53. So this is a pretty important transcription activation complex. So the target genes include genes that are involved in stopping the cell cycle, genes that are involved in controlling apoptosis, genes that are involved in controlling DNA repair. So a lot of target genes can be activated. One of the target genes is called MDM2. And the consensus sequence located in the promoter of MDM2 has a high affinity for P53. So even if you have low quantities of P53, you will first go on MDM2 promoter before other promoters. And MDM2 is a protein that, once made, is able to bind to P53 complexes, and it will flag it for proteasome degradation by the means of ubiquitin chains. So basically, P53 is produced and very quickly assembles in a complex, very quickly moves on the promoter of MDM2. MDM2 is produced in the form of a protein. The protein MDM2 flags P53 for degradation. P53 is degraded. As a result, you never have a strong buildup of P53 in the cell. Now, in order to do that, MDM2 needs to be phosphorylated, and it is phosphorylated on an activatory site. And this activatory site will trigger P53 inactivation or P53 degradation. One of the most important enzymes that will phosphorylate MDM2 on its activation site in order to promote survival signals is AKT, also called PKB sometimes. With P53 degraded, you lift some sort of control on the cell cycle. On the other hand, if you have a damage to the DNA, single-strand DNA damage or double-strand DNA damage will all result in the activation of sensors that will activate kinases like ATR, ATM. These kinases will activate another final kinase called CHIK1, and CHIK1 will produce the phosphorylation of P53. So from a not very active and prone to degradation P53 complex, we now switch to an activated P53 complex that has a higher affinity for its target genes, and it will then be able to activate other genes than MDM2. If you don't have the survival signals, then MDM2 will also be targeted by CHIK1. So CHIK1 will phosphorylate P53 and it will also phosphorylate MDM2. However, this time, instead of phosphorylating MDM2 on its activation site, it will, in contrast, phosphorylate MDM2 on an inactivation site. So at this point, MDM2 is no more able to bind with P53 and can not inactivate P53. So when the damage is sensed by the sensors, CHIK1 promotes activation of P53 and inactivates MDM2. Then P53 that is being constantly produced will start to build up instead of being degraded. When the quantity of P53 complexes is increased, they will now be able to bind on a lot of other target genes. The first and most highly affin for P53 are P21, which is a cyclin inhibitor, GAD45, which is a DNA repair activator. So survival signals will increase the production of MDM2 and phosphorylate it in its activation site to remove P53. DNA damages will, in contrast, increase the phosphorylation of P53 to increase its activity and increase the inactivation of MDM2 in order to avoid degradation of P53.